Welcome to EPG Pathshala. I am Dr. Kamal Kumar Gupta, Associate Professor in Zoology at Deshbandhu College, University of Delhi. This is a module on transgenic animals applications. This module is included in the paper Molecular Genetics. After studying this module, you will learn about transgenic animal has been widely used in research for understanding biological basis of genetic and infectious disease. Many transgenic animals such as glowfish, aqua advantage, salmon and others have been created and approved for human use. Knockout mice project aim to produce mouse with knockout mutation in the genes in the mouse genome. Transgenic livestock have been used and approved for production of pharmaceuticals and therapeutics. In future, transgens can be used in production of disease-resistant livestock, environmental friendly organism, organ for xenotransplantation and monoclonal antibodies, etc. Let's start with the introduction. Transgens has wide applications in improving genetic features of domesticated animals and production of human pharmaceuticals in the farm animals. Transgenic animals are suitable model for study of human disease, study the gene regulation, tumor development, immunological specificity and molecular genetics of development of animal can also be studied on transgenic animals. Transgenic animals can be used as bioreactor to produce useful pharmaceutical products. Some of the major thrust areas in application of transgenic animal include enhancing nutritional value of milk, protecting farm animals against common pathogens that cause disease and animal loss. Now, we will study application of transgenic animals. Transgenic animals are suitable model for determining biological basis of disease. They reveal some potential targets for their treatment. Transgenic mouse models have been developed for human genetic disease such as Alzheimer disease, Huntington disease, arthritis, muscular dystrophy, hypertension, neurodegenerative disorders, endocrine dysfunctions and coronary disease, etc. Transgenic mouse model have been developed for Alzheimer disease. You may be knowing that Alzheimer disease is a chronic neurodegenerative disorder that is characterized by dementia, the progressive loss of abstract thinking, memory and intellectual abilities. It results in personality changes, language disturbance and a slowing of physical capabilities which interfere with the daily life. The patient show accumulation of neurofibrillary tangles within the cell bodies of the neuron. Development of dense extracellular aggregates called senile plaques at the end of inflamed nerves and loss of neuron in the neocortex and the hippocampus region of the brain. The core of senile plaque is composed of structure called amyloid body. The amyloid body contain a beta protein which are derived from an internal proteolytic cleavage of the beta amyloid precursor protein, also called as APP. Faulty cleavage of the APP protein causes the production of A beta 40 and A beta 42, the main variants in the Alzheimer disease. Inefficient clearance of the alpha amyloid likely leads to their accumulation. Now, mouse model for Alzheimer disease were created with transgene that contain mutation in the APP gene. Two mutant gene of APP, that is APP 717 and APP 670-671 were used for creation of transgenic mouse model for Alzheimer's disease. A transgene with the APP717 mutation was constructed from an APP cDNA. Modified introns were added between exons 6 and 7 and 7 and 8 and 8 and 9 of the APP cDNA. Presence of intron in the transgene increases the rate of transcription. The promoter sequences from platelet-derived growth factor which express in the brain were taken for APP cDNA intron construct. The complete construct is called the PDAPP minigene. The transgenes were introduced in the mice. The transgenic mice containing about 40 copies of PDAPP minigenes on aging displays amyloid plaques, neuronal cell death and memory defect. Mice with APP 670/671 gene construct also produces Alzheimer's disease like features. The formation of amyloid plaques in human has also been shown to be associated with increased production of a protein BACE1, that is beta site APP cleaving enzyme 1. Protease that cleaves APP to produce A beta. Reduced production of BACE1 by RNAi, that is RNA interference, is a possible way to reduce or delay Alzheimer's disease. shRNA that target BACE1 mRNA were carried on a lentivirus vector that was injected into the hippocampus in transgenic mice. These mice showed a reduction in the A beta deposits and plaque formation. Now, we will be coming to the next disease, that is transgenic mice model for the Huntington disease. You know that Huntington disease, an incurable fatal neurological genetic disorder, it remains confined to a specific region of the brain. About 1 in 10,000 people worldwide are affected by this disease. The gene is due to change in the disease is due to change in the HD gene code the protein Huntington. It has been seen that addition of CAG trinucleotide unit to the exon 1 of the HD gene is responsible for the disease. During translation, CAG code for glutamine, consequently multiple CAG codon incorporate a series of glutamine residue that we also called as polyglutamine in the Huntington protein. Symptoms of Huntington disease occur when number of CAG codon in polyglutamine region is 38 or more. A mouse model for Huntington disease was created using TET of conditional regulation system. HD gene that contain exon 1 with 94 CAG repeats were used as the transgene. The TTA genes was placed under the control of promoter that is active in the cells of the forebrain. Expression of HD transgene was switched off in the embryo during pregnancy by adding doxycycline to the drinking water. After birth, doxycycline was not supplied to the transgenic mice. This allowed continuous expression of mutant HD gene and the production of protein with a long polyglutamine sequence, a neurological condition that was similar to Huntington disease in human was developed in these transgenic mice. The feature of the disease disappeared when the expression of mutant HD gene was prevented by addition of doxycycline. This indicates that a continuous expression of a mutant HD gene is required for establishment of the disease. The brain cells can recover when the synthesis of HD protein ceases. Transgenic primate model such as rhesus macaque have also been developed for such human neurodegenerative disorder for better understanding. Transgenic mouse model for infectious disease. Many of the infectious disease models has also been created in mouse. Pseudorabies virus is an alpha herpes virus that infects pigs. Viral infection results in encephalitis and respiratory illness in the young pigs and abortion and infertility in the sows. Nectin-1 is a porcine receptor to which alpha herpes virus binds. Entry of the virus into the host cell can be blocked by expressing a soluble form of this host cell receptor, that is nectin-1. This would prevent this virus from binding to the host membrane bound receptors, hence prevent viral penetration of the host cell. Before the creation of transgenic farm animals, it was tested for its ability to protect against pseudorabies infection in a mouse model. A transgene was constructed from the DNA sequences encoding the extracellular domain of the nectin-1. This was fused to the gene for the constant region of human immunoglobulin G, that is IgG. It was placed under the control of a promoter so that it can express in several type of cells. This fusion construct could produce a secreted form of the nectin-1. The transgenic mice were found resistant to the pseudorabies virus. In this way, transgenic pigs can be produced with resistance pseudorabies virus infection. Transgenic mice are also used as model to study effect of cell death. In human beings, diphtheria toxin is produced by the bacterial pathogen Carinobacterium diphtheria. It binds to the heparin binding epidermal growth factor receptors present on the cell. The toxin receptor complex is then taken up into the cell where it inactivates elongation factor EF2 required for the protein synthesis. Failure of protein synthesis leads to cell death. The cell death can be induced in transgenic mice in order to study organ failure resulting from cell death. Mouse cells are not normally susceptible to diphtheria toxin because they do not have a receptor that recognizes the bacterial protein. Transgenic mice were engineered to express the human heparin binding epidermal growth factor receptors under the control of liver specific promoter. This results in expression of HBEGF receptors in the cell membrane of the liver cells. Treatment of these transgenic mice with diphtheria toxin leads to liver damage. The transgenic animals have a variety of medical applications. They can be used for production of pharmaceuticals. The mammary gland of the dairy cattle can be used as a bioreactor for the production of pharmaceutical proteins and therapeutic agents. Many transgene constructs that have mammary gland specific promoters and human gene sequence has been successfully introduced and expressed in the milk of transgenic sheep, goats, pigs and rabbits. The advantage of the transgene derived protein is that these proteins are glycosylated and have other post translational modifications. The protein secreted in the milk usually have biological activities similar to the human protein. To express the human hormone gene in the cattle milk, the coding sequence of the human hormone gene is linked with the promoter of beta lactoglobulin gene. A gene is normally expressed in the mammary gland cells. In addition, a short signal sequence is necessary for protein secretion in the milk was also included in the transgenes. These transgenes are incorporated in the sheep. In this way, transgenic sheep producing human hormone in their milk can be created. The hormone can be purified from the milk and used to treat humans. Transgenic cattle have wide application in production of pharmaceuticals and therapeutics. High yielding varieties of cattle can produce approximately 10,000 liters of milk annually. If amount of recombinant protein in the milk is 1 gram per liter and it could be purified with 50% efficiency, then 20 transgenic cow would be yielding about 100 kilogram of the recombinant protein per annum. This much yield suffices the annual global requirement for protein G, which is used for prevention of blood clots. Similarly, 1 transgenic cow would be sufficient for production of the annual world supply of factor 9, that is plasma thromboplastin component, which is required and used by hemophilics to facilitate blood clotting. Transgenic goats and sheep can also be raised to produce pharmaceuticals in their milk. Recently, US Food and Drug Administration approved the human protein antithrombin produced in the transgenic goats milk for the use in the individual with a hereditary deficiency of this protein. The milk of transgenic goat is a significant source of human antithrombin, which yield 2 to 10 grams per liter of milk. It has been estimated that 75 transgenic goats are sufficient to meet the annual worldwide demand for antithrombin. Many other human therapeutic proteins such as antitrypsin, human clotting factor, factor 9 for the treatment of hemophilia and monoclonal antibodies have also been expressed in transgenic goats. Transgenic animals have been developed for production of fully human monoclonal antibodies. Recombinant DNA strategies have been devised to humanize existing rodent monoclonal antibodies to create a transgenic mouse that is capable of synthesizing a full range of human antibodies against every antigen. The endogenous mouse heavy and light chain genes were inactivated and YAC carrying most of the heavy and light chain DNA element from each human immunoglobulin gene were inserted into the chromosomal DNA of the mouse. A commercialized version of the human antibody producing mouse has been designated as Xenomouse. The first fully human monoclonal antibodies produced in the mouse has received regulatory approval for use as treatment for the advanced colorectal cancer. Other therapeutic antibodies produced in the Xenomouse include several for the treatment of various cancer and osteoporosis are now in clinical trial. Donor organs can also be produced in the transgenic animal for xenotransplantation. Organ transplant is recommended in the case of organ failure. Currently, organs such as heart, liver, kidney are taken from donor and transplanted in the recipient. However, demand for donated organs far exceeds than the available supply. Animal to human transplant, that is xenotransplantation, can be a way to supply organ for transplantation. Pig organs can be considered for xenotransplantation as they are similar in size and physiological function to those of human. A major limitation of xenotransplantation is hyperacute rejection of the animal organ by the recipient. It is due to binding of the pre-existing antibodies of the recipient to a carbohydrate epitope, that is alpha galactose, present on the surface of the cell of the transplanted organ. This elicits an inflammatory response that destroys the transplanted organ. It was proposed if the donor animal carried one or more of the gene for a human complement inhibiting protein, a transplanted organ would be protected from the initial inflammatory response. Transgenic pigs with different human complement inhibitor genes have been produced. Hyperacute rejection did not occur in the primate recipient after kidney from transgenic pig was transplanted. Survival period was 20 to 90 days. Another strategy for xenotransplantation is to produce transgenic pigs with the organs that do not produce the antigenic alpha galactose epitope by deleting the gene encoding 1,3 alpha galactosyl transferase. Transgenic animals can provide improved nutrition quality. Transgenic cattle with improved nutrition value of milk for human and for the suckling have been produced. Overexpression of protein in the milk can improve the growth, health and survival of suckling animals. Specific components of milk can also be altered as per human requirement. Cheese production from milk is directly proportional to the beta casein and K casein contents. The amount of these proteins can be increased in the milk of the cows engineered with additional copy of these proteins. Lactose intolerance animal lack lactose hydrolyzing enzyme lactase and therefore experience severe indigestion after the consumption of milk. Decrease in the lactose content of milk can be achieved by expression of the mammalian lactase gene in the mammary glands. Many people are allergic to the beta lactoglobulin present in the bovine milk. Knockout mutant of beta lactoglobulin can be created to remove beta lactoglobulin in the milk. Similarly, omega 3 fatty acids content can be included in the pig. Omega 3 fatty acids are the long chain polyunsaturated fatty acids found mainly in the fishes. Human and other livestock animals cannot produce these fatty acids. The livestock animals contain high level of omega 6 fatty acids. They cannot convert omega 6 fatty acids to the omega 3 fatty acid as they lack the enzyme desaturase. Diets with high omega 6 fatty acids can cause many diseases such as cancer, heart disease and diabetes. Transgenic pigs that synthesize omega 3 fatty acids has been produced. The roundworm Xenotransplantation elegans produce an enzyme desaturase that converts omega 6 fatty acids to the omega 3 fatty acids. A transgene was created containing enzyme desaturase gene, that is fat A, from Xenotransplantation elegans. The gene was cloned into an expression vector. Fetal pig fibroblasts were transfected and cultured. The cultured cells that produce higher level of omega 3 fatty acids were used to produce transgenic pigs by nuclear transfer. The transgenic pigs showed threefold higher level of omega 3 fatty acids and 23% lower level of omega 6 fatty acids than the non-transgenic pigs. Many of the transgenic animals are environment friendly. EnviroPig is one such example. This is a trademark for genetically modified line of Yorkshire pig with the capability to digest plant phosphorus more efficiently than the conventional unmodified pigs. These transgenic pigs were developed at the University of Guelph. The main food source for the pig is soybean meal, which has about 50% or more of its phosphate in the form of phytate. The pigs are unable to digest and utilize the phytate due to absence of the enzyme phytase. Therefore, they excrete large amount of phosphorus, which causes serious environmental problems. The enzyme phytase is found in plants and many microorganisms, which remove phosphates from phytate. A transgene consisting of phytase gene, that is APA, from E. coli and the parotid secretory protein promoter was constructed. Transgenic pigs were created by pronuclear microinjection. The transgenic pigs produces the enzyme phytase in the salivary glands, that is secreted in the saliva. This phytase digests phytate present in the food. Another environment friendly animal is transgenic Mastitis fish. This was created for pollution monitoring. Synthetic derivatives of natural estrogen are used in most oral contraceptives and also as a therapy for post-menopausal disorders in women, also to treat infertility and endometriosis, and to develop female only fish population in the aquaculture. A wide variety of industrial chemicals such as bisphenol A and polychlorinated biphenyls, that is PCBs, also have estrogenic activities in animals. A large amount of these estrogenic chemicals are flushed into aquatic ecosystem with domestic, agricultural and industrial wastewater and cause water pollution. Transgenic Mastitis has been developed to monitor quality of water and detect estrogenic compounds in aquatic environment. A transgene was constructed for green fluorescent protein under the control of estrogen responsive promoter from the Mastitis vitellogenin gene. The gene was cloned and injected in the Mastitis eggs. Exposure of transgenic fish to 17 beta estradiol and other natural and synthetic estrogenic compounds activates the vitellogenin promoter. This leads to production of green fluorescent protein that can be visualized as emission of green fluorescence in the living fish. A variation of this transgenic model can be used for the assessment of heavy metal contamination of water. A heavy metal inducible promoter can be incorporated adjacent to the red fluorescent protein gene. This transgene can be used to create transgenic zebrafish. When these transgenic zebrafish are kept in water contaminated by mercury and other heavy metals, the promoter become activated and induce expression of the red fluorescent protein gene. Transgenic animals which are resistant to infectious disease have been developed. Mastitis, that is a mammary gland abscesses in dairy cattle, bovine spongiform encephalopathy, that is BSE, also known as mad cow disease in cattle, and neonatal sores, that is dysentery in swine, are some of the target disease for production of transgenic disease resistance livestock. The approach for this is production of disease resistance cattle included in vivo immunization, in which transgene encoding the heavy and light chain of monoclonal antibodies was introduced into the recipient animals. Expression of a monoclonal antibodies against a specific pathogen provide immediate protection without prior exposure to the pathogen. If the monoclonal antibodies is secreted into the milk, young suckling animals also acquire passive immunity against a pathogen. Another approach is to produce disease resistance livestock is the elimination of the host cell component to which the infectious agent interacts. Bovine spongiform encephalopathy BSC, also known as mad cow disease, is a neuropathological disorder in cows. The brain tissue of infected animals become filled with holes that gives the brain a characteristic sponge-like appearance. It is caused by a mutant form of the prion protein. The mutant prion protein induce the brain protein to misfold. The misfolded protein aggregate and disrupt normal brain functions. There is no known treatment for this disease, and therefore, the infected animal have to be destroyed. Moreover, the prion can be transmitted to human through consumption of prion contaminated meat and can cause a variant form of encephalopathy in human being. In transgenic cows, both the alleles of the gene encoding normal form of prion protein were knocked out by the insertion of an antibiotic resistance gene into the coding sequences. The genetically modified animals were found normal for a variety of morphological and physiological features, including mental status, sensory and motor functions, immune functions, and brain tissue morphology. When brain tissue homogenate from wild type and knockout cattle and incubated with brain homogenate of BSC infected cattle, propagation of BSC prion could not be detected in the homogenate of knockout animals. Mastitis is an infection of mammary gland of cows. It can block milk duct, reduce milk output, and can also contaminate the milk with pathogenic microbes. It is caused by the bacteria Staphylococcus aureus. These infections are contagious and readily spread in the entire herd. In an attempt to create cattle resistance to mastitis, transgenic cows were generated that possessed the lysostaphin gene from Staphylococcus. Lysostatin is an enzyme that specifically cleave components of the Streptococcus aureus cell wall. A transgene consisting of altered lysostaphin gene under the control of bovine beta lactoglobulin promoter was introduced into cow fibroblast. The nuclei from these cells were then transferred to enucleated oocytes and activated. Blastocysts were transplanted into the uterus of cow and several calves were subsequently born. Transgenic cows expressing this protein in the milk provide immunity to the suckling against Staphylococcus aureus infection. Transgenses could be used to improve the genetic makeup of chickens with respect to the disease resistance, with respect to the lower fat contents and cholesterol level in the eggs and better meat quality. The eggs with its high protein content could be used for production of pharmaceutical protein. A transgene can be expressed in the cell of reproductive tract under control of oval albumin promoter and regulatory elements. This can yield up to 1 gram of recombinant protein in the eggs. Transgenic chicken that synthesize monoclonal antibodies, growth hormone, insulin, human serum albumin and alpha interferon have also been produced in the transgenic chicken. Enhanced growth rates, tolerance to environmental stress, resistance to the disease and model for pollution monitoring are some of the traits considered for the creation of transgenic fishes. Transgenes was created consisting of promoter region and polyadenylation signals from the antifreeze protein gene of ocean pout and growth hormone cDNA from Chinook salmon. These transgenes were injected into the eggs of Atlantic salmon and aqua advantage salmon were created. Presence of promoter from the antifreeze gene resulted in expression of growth hormone in cold water. Hence, the transgenic salmons were larger and grow faster than the non-transgenic fishes. Conceptually, the faster growth of farmed salmon would lower the cost of feed and lessen the pollution of coastal water. On November 25, 2013, Environment Canada approved the product for salmon egg production for commercial purpose in Canada. In May 2016, the Canada Food Inspection Agency approved the sale of the GM fishes. Scientists at Yorktown Industry of Austin, Texas created the glowfish, a transgenic strain of zebrafish containing a red fluorescent protein gene from sea anemone. Glowfish fluoresces bright pink when illuminated by ultraviolet light. It was marketed as first GM pet in the United States. A variety of different colored glowfishes are currently available at the pet stores. Now, let us summarize the module. Transgenic animals has wider application in the field of research, production of pharmaceuticals, therapeutics, development of disease resistance livestock, production of environment friendly animals and improving nutritional quality of the animal products. Transgenic mouse models have been developed for human genetic disease such as Alzheimer disease, Huntington disease and many others. Transgenic cattle can be used for production and secretion of pharmaceuticals and therapeutics in their milk. Human protein antithrombin produced in the transgenic goat milk has been approved by US Food and Drug Administration for the use in individuals with hereditary deficiency for this protein. Xeno mouse is a transgenic mouse, produces fully human antibodies. First fully human monoclonal antibodies produced in the Xeno mouse has received regulatory approval for the use as a treatment for advanced colorectal cancer. Transgenic animals can produce organ for xenotransplantation to human being. This may be achieved by inserting human complement inhibitor gene or deleting alpha galactose epitope by knock down the gene encoding 1,3 galactosyl transferase in the transgenic animal. Transgenic animals with improved nutritional quality has been produced, increased casein quantity, secretion of the enzyme lactase in the milk and knockout of beta lactoglobulin gene are some of the desirable trait in the milk. The gene for enzyme desaturase from Caenorhabditis elegans is expressed in the transgenic pigs. This gene can convert omega 6 fatty acids into unsaturated omega 3 fatty acids. EnviroPig is environment friendly pig. It contains a transgene for enzyme phytase. The enzyme phytase is secreted in the saliva and can digest phytate present in the feed. This decreases the phosphorus content in the excreta of the transgenic pigs. Transgenic medaca containing gene for green fluorescent protein can be used to monitor estrogenic compounds in the waste water bodies. Transgenic animal resistance to infectious disease such as mastitis, that is mammary gland, abscesses, bovine spongiform encephalopathy, that is mad cow disease, and neonatal sores, that is dysentery, have been developed. Transgenesis can be used to improve the genetic makeup of chicken with respect to resistance to the disease, lower fat and cholesterol level in the eggs, and better meat quality. Pharmaceuticals can also be produced in the egg of transgenic poultry. Aqua Advantage salmon contains a transgene construct with promoter of antifreeze gene from ocean pout and growth hormone sequence of Chinook salmon. These salmons are larger in size and grow faster than the non-transgenic salmon. Glowfish is the first GM fish containing gene for fluorescent protein. Different type of glowfish are available at the pet stores. And here we end the module. Thank you.